So once again, I'd like to thank Esper and the organizers for inviting me to talk this afternoon. I'm going to look at the data in a little bit different way and take a position that, or offer a position about the cure agenda that maybe is a little bit more controversial. Steve talked about we have to understand the pathogenesis of HIV, especially the kinetics, if you will, of the reservoir, however you define it, but we have to understand that during treated infection if we're really going to try and come up with a rational approach to a cure. And so I'd like to walk through some data that we and others have generated and put it together in a slightly different way and think about the possibility that the drugs that we're giving today, for the most part, are not doing what we think they're doing in the lymphatic tissues where 99.6% of the virus is. And so I'd like to walk you through that data and see if I can convince you that maybe we need to think about this in a slightly different way. So when I'm thinking about the cure, I'm reminded of this quote from William Shakespeare. The task he undertakes is numbering sands and drinking oceans dry. It's a very large task that we've given ourselves to think about how we can completely cure people from this infection, as Steve said. And we are continuing to have the discussion about what is the actual goal, what do we need to be doing. So you've heard a lot about the Berlin patient from Steve, and I'm just going to focus on, we think that he was cured, and what are the critical components that led to that cure, so we know what we have to duplicate in our strategies for eradication. The first thing that they did with his chemotherapy is that they cleared the reservoir of infected cells. They activated some virus from latency, both with the chemotherapy drugs and the radiation, but also with the GVHD, the graft versus host disease, that he had as a consequence of the transplant, both transplants. But critical and key to his success, I think, was that they replaced his cells with cells that couldn't be infected with HIV. So with the Delta 32, the cells from a donor that lacks CCR5. And those three elements, I think, were the critical elements that led to the success with him in particular. Now, we've had some other attempts at cure over the years that have not been successful. And so what I'm showing you on this graph is the viral load in copies of RNA per mil of blood, and then the time to rebound. So this is a treatment interruption graph, if you will. And the red here is Timothy Brown, the Berlin patient. And he was completely undetectable, still out many years after his transplant. Here, and this, I'll show you this data in a moment, but this represents people in chronic HIV infection. So this is our data from a few years ago. When they're started in therapy after they've been infected for a while, months to years, and you treat them for a while, in this case a mean of 10 years, and you interrupt therapy, the virus rebounds in 14 days on average. The range was 5 to 30. So rapid recrudescence of viremia when you stop therapy in that setting. Now, here you have the Boston patients, the bone marrow transplant patients. What they had was everything that Tim Brown had, except for the Delta 32, for the CCR5 negative marrow cells replacing. And those patients went off after the transplant and prolonged suppression, went off therapy, and 10 to 14 months later, both of them rebounded. Now, the thought here was that, by giving them therapy, you eradicated the reservoir, and that turned out not to be true. We also have experience with the Mississippi baby. So this is the infant that was treated at the time of birth when it was discovered that mom was HIV infected, and RNA tests at the time of birth showed that the baby was getting infected as well. So at that time, they started therapy, and the logic there, the strategy would be to either prevent the infection or dramatically reduce the size of the reservoir so the immune response can be more robust so that you can hopefully prevent that virus from rebounding. So this baby was started on therapy, disappeared from treatment, I forget for how long, came back in. Therapy had been stopped for a variety of reasons, and that child rebounded approximately 30 months later. So The difference there is that here we thought we had reduced the size of the reservoir, and probably did. Here we did not give CCR5 negative cells. And so in these cases, we have not recapitulated, if you will, what had been accomplished with Tim Brown. But we did see a significant delay in recrudescence, and I think that's important. Oops, wait a minute. Okay. All right, so the strategy then, or the idea is that HIV infection is characterized by high levels of circulating viruses in the blood. You start antiretroviral therapy. You see this viral load come down to undetectable to under the limit of detection. Although if you were to use a single copy assay or some other very sensitive technique, in many people you would still find evidence of HIV RNA in the blood, but it's below the critical threshold here, in this case of 30 copies per mil or 20 copies per mil. So that's what happens when you give antiretroviral therapy. And then in every patient that I know of, save one, Tim Brown, at some point after you stop therapy, the virus rebounds. And the question that I have, and the thing that I'm most interested in, is where does this come from? What is the source of this recrudescent viremia? And why is it different for different groups of people depending on the intervention that you use? Because there's no question but that it is. So where does this come from? So this is our treatment interruption data, where I told you that within 14 days, a mean of 14 days, 2 weeks, most people rebound. And what you see here is the viral load per copies per mil. And this was the day that the virus was first detected. And this is when we came back and actually did the biopsies that I'll show you later in the talk to characterize the source of recrudescence. But this is just to give you the idea that this happens rapidly and always within 30 days. Now, this is data that was recently, or it's in press, I guess. And so what we've done is overlaid our data from the chronic infection with data from the RV254 study. So this, as you know, is the acute infection study in Bangkok, where patients are identified in one of the FEBIG stages and immediately started on therapy. They had several patients who were in FEBIG 3 and 4 when the patient was started on therapy. Recall, FEBIG 3 is when antibodies are detected. It's typically when you would see the onset of symptoms of seroconversion. So FEBIG 1 and 2 is the ramp up, the rapid increase of virus in the blood, and then you begin to see antibodies, and FEBIG 3 is when you have a more full evolution of the antibody profile. And so this is FEBIG 3 and 4, but the point is that this is the earliest time that you could be giving antiretroviral therapy. So not like the Mississippi baby, where she was started on therapy at the time of exposure, which is more like what Steve was talking about with the patient who was given PrEP, although I think the Mississippi baby was even earlier. But these patients were started in FEBIG 3 and 4, and the median time to recrudescence was 22 days. So this is another example of where you limited the reservoir, the size of the reservoir, yet you did nothing significant to delay that time to recrudescence, okay? So the question remains, where is this coming from? And we know that very early therapy is, at least as defined by this measure, is not impacting the kinetics of the recrudescence in any meaningful way. Well, in those same patients, now this is a large group of patients from the Bangkok study, where we were able to get lymph nodes at FEBIG 1, FEBIG 2, FEBIG 3, and FEBIG 4 stage of infection, and using in situ hybridization for HIV DNA, we can visualize and count the number of cells that have HIV DNA in them. So this is total DNA. We're not doing any molecular methods here to say, is that integrated, you know, et cetera, et cetera. This is just the cell has DNA in it. And even by FEBIG 1, so, you know, during the ramp up, even by FEBIG 1, you see that there's this large number of cells per gram of lymph node tissue. There's a large number of cells that have already established infection. And it goes up by about a half a log in FEBIG 2 and stays steady by FEBIG 3 and 4. And I'll point out that this is exactly the same number that I see in lymph nodes that I collect from individuals in Uganda who have been infected for many years. and then we take a lymph node and we look, or patients in Minnesota who are who are infected but not on therapy. This is, at least by the measure that we use in situ hybridization, this is about what is established in chronic infection, and that occurs already by FEBIG2. So it's a rapid establishment of that reservoir. So I'm putting in here now the data from Bangkok to show that even when you limit the size of the reservoir, you still have this rapid recrudescence. Okay, so the question then is, what are the cellular sources and anatomic sites of virus recrudescence when treatment is interrupted? Now, a few minutes ago, I made the statement that the reservoir is primarily in the lymphatic tissues, gut, lymph node, spleen, secondary lymphatic tissues. And I'd like to show you a little bit of that data to convince you. Let me back up for a second. So the question is, what are the cellular sources and anatomic sites? And there are two sort of competing views on this. The first is the predominant view, that when the virus comes back, it's primarily reactivation from latency. And the model here is that there is one or just a few cells that reactivate virus, that you have rapid expansion, dissemination, and that's what's driving the recrudescence. So what you have is a latently infected resting central memory CD4 cell. Something comes along to activate that cell, and then you have virus being produced from that activated cell, and that's an important point, activated cell. So the data that support that are that there, in all but one study, there's been no evolution of virus sequences, and this is typically in peripheral blood while on antiretroviral therapy. So sequencing doesn't seem to support that. And there's a lot of debate in the field, of which I am not a member of the evolutionary biology field, but the debate that I hear is differences in methods used to sequence, different populations, et cetera, et cetera. So but it is a point that is made. There's no emergence of drug resistance. That's an important point and one to consider. And then, with the exception of one study that I'm aware of, there's been no change in HIV DNA, cell-associated or plasma HIV RNA with intensification. So these are all really compelling arguments arguing against or arguing for the model of reactivation of latency and against the model that says there's persistent production or replication of virus while on antiretroviral therapy. The problem is that these studies all have only been done in peripheral blood. And that's a real limitation because there is a thought out there that the blood is the window to the soul, if you will, that what happens in the blood is absolutely representative of what happens in tissue sanctuary sites. And I do not believe that's the case, and I'll show you some data to suggest that, but I think that if you're really going to study this, you have to go looking in the lymphatic tissues. So the other view is that the source of recrudescent viremia is ongoing low-level infection of new cells despite the presence of antiretroviral therapy. So you have these cells that continually produce virus, and that this resting T cell is somehow infected and perpetuates or is one of the reasons why this is a persistent infection. This is a very important, critical question in my view, because if it's only latency that's the issue, well, then we think about this path for cure. But if there's ongoing low-level production, then you've got to think about another strategy altogether, because until you deal with that, the latent pool is going to be the latent pool. So if it's latently infected resting memory CD4 cells, then Steve had talked about shock and kill, or keeping the virus dormant so that it doesn't come out of latency. And there's a whole pathway for thinking about those cure strategies with HDAC inhibitors, et cetera, et cetera. However, if this is the source of recrudescent viremia, then you have to optimize antiretroviral therapy and come up with adjunctive therapies to achieve full suppression of virus replication in the lymphatic tissue reservoirs. And I want to point out and stress, and I'll repeat this, that this model does not preclude this model. In fact, I believe both things are an issue and both things need to be addressed. So the question to answer about the HIV reservoir is how do we define it? And I think if you lined everybody speaking up at this conference, we'd all have a slightly different take on that. Where is it? I'll show you a little bit of data on that. How complex is it? This issue of, is it purely latent virus? Is that important? Is it ongoing replication? What's the mix? And then how big is it? What is the size of that reservoir? So this is the way that we look at it, and some of you have seen this slide or slides similar to this before. This is HIV RNA in situ hybridization. And what you're looking at is a lymph node from a patient with chronic HIV infection who has never had antiretroviral therapy. And what you see here is the B-cell follicle, and you see all of these green dots are virions that are attached to the follicular dendritic cell network, which you heard about earlier, the FDC network, by immune complex. And that virus is made in the parafollicular T cell zone by these cells, which light up. These are RNA positive cells. And so the virus that's made there shows up on the FDC network here. Now there's one exception to that, and this is a really interesting part of the pathogenesis, is that the T follicular helper cells in the B-cell follicle are also an engine of production, and it is possible that a lot of the virus that you see here are made by the productively infected cells in the T cell zone, which are inevitably T follicular helper cells. But whatever, what you're looking at here are probably three distinct reservoirs. This reservoir out in the parafollicular T cell zone where CD8 cells can work, etc. The T follicular helper cells in the germinal center and the B cell follicles where the CD8 T cells can't get to. And then the virus that is on the FDC network that is persistent and stays for a very long time. And that virus can come off of the FDC network and go on to infect new cells. So at least in this cut, in this slice, we're looking at probably three distinct important reservoirs prior to treatment of antiretroviral therapy. And the way that we measure that and the way we look at that was relatively simple quantitative image analysis techniques. So here you have the cartoon that you have infected, activated CD4 T cells making a lot of virus that gets trafficked into the FDC network. You have these infected resting CD4 cells that I'll talk a little bit more about in a moment, but these are Ki67 negative cells that do make virus that also is represented on the FDC network. And here by in situ, you can see in a tissue slice, in a tissue section, these individual positive cells. And so what we can do, very simple, is we can just count the number of these cells, depending on the area that you're looking. We know the area of the tissue, and so we can express these as number of cells per gram of tissue, and we can get to grams because we know the density of the tissue, right? So we can now express this as number of infected cells, whether it's RNA, DNA, et cetera, per gram of tissue. But we've also worked out methods where we can count the number of viral particles on the FDC network. And this turns out to be incredibly important because one of the measures that we use to look at effectiveness of antiretroviral therapy is the rate of decay off of this network. Because papers that a colleague of mine, Ashley Haas, published in the late 90s showed that the rate of deposit onto the FDC network is fairly equal to the number coming off at any given time. So if you've got a steady state production, this will stay constant. If you somehow reduce with antiretroviral therapy, you can measure the rate of decay, and that gives you an idea of another measure of efficacy of the drug. Jake Estes, who many of you know, took this technique. This is with a radio-labeled probe. It's a fussy assay. It can go down easily for a while. It takes a long time to do. Jake developed another alternative way of doing HIV RNA in situ hybridization that's much more facile. We can get it done more rapidly and apply the same image analysis techniques to it. So we've got these tools that we can characterize the reservoir as I've just defined it. So first I'm going to show you that this is located mainly in the lymphoid tissues. And what I'm showing you in this cartoon is the FDC network in both gut-associated lymphoid tissue and the lymph node. And then you've got these infected resting cells, activated cells making virus that traffics into the germinal center, but also is the source of viremia in peripheral blood. And that is a point that I think we sometimes lose sight of. There are very few, and we've done this, if you count the number of protectively infected cells actually making virus in peripheral blood, they're relatively few and does not account for the amount of virus that you see in peripheral blood. So the virus that you're measuring in peripheral blood is coming from somewhere. other than the peripheral blood. And I'll show you why we think this is coming from the lymphoid tissues. So this is a paper we published in Nature Medicine several months ago now. What we did was we took monkeys that had been infected, who were not on therapy, and we took all the organs and we systematically went through and looked at the frequency of productively infected cell, RNA-positive cell per gram of tissue, and just expressed it here in this map. We looked at lymph node, gut, spleen, brain, kidney, heart, lungs, liver. And this is just to show you that 99.6% of the RNA-positive cells prior to antiretroviral therapy are in lymphoid tissue, 35% in the lymph node, 62% in gut. When you go through and you do the image analysis, counting the number of virions that are on the FDC network and the number of RNA-positive cells, these are the kinds of numbers that you get. So it's a fairly large reservoir that we're talking about. But we need to be looking at this at higher resolution, at least with the techniques that we're using. And there's another method of in situ hybridization with tyramine signal amplification using a DIG probe. And so when you do that, you can actually visualize the virions, the viral particles that are coming from the cell. And that's what this looks like. So here's our sense probe, and here are, this is in situ hybridization for HIV RNA using this TSA-ELF method with the DIG probe. And so you can appreciate right away, you can see these clouds of viral particles that are coming out of the cell. Some of the cells are quite large, some of the cells are small. The cells that are large have a large cloud of virions. The cells that are smaller have a smaller cloud of virions. And if you do just a simple Ki-67 stain, which, in my world of histology, we only get a few markers to look at stuff. But if you do the double label with the TSA-ELF and the Ki-67, you'll see that all of these large cells are activated cells that are making a lot of virus. And then you have the small resting CD4 cells that are Ki-67 negative, that just have a few virions coming from them. And this is a paper that Ashley Haas and Kevin Riley published in 2007, showing that when you count the virions that are coming off the cell, whether they're Ki-67 positive or negative, you'll see that there's two distinct populations. There's this activated population that has, on average, about 200 virions. This has a half-life of 1.5 days, as Rick pointed out earlier. But there's also a population of cells that are small, Ki-67 negative, they're resting cells, and this is one of the pieces of evidence that really showed, or when we started to talk about the idea that resting CD4 T cells can be productively infected. They make on average about 35 virions in a snapshot, and their half-life is 4 to 14 days, so a much longer population of cells. And so when you go through and you start looking in acute, early, or chronic, and just say what population is sustaining the infection in untreated people, in acute, and it's again, it's primarily the activated cells. As time goes on, this becomes the dominant population of cells that are sustaining the infection. So I just want to cement this idea that there are two things that are producing virus that occur in the follicular dendritic cell network that we use to measure the efficacy of the drugs, the decay rate off the FDC network. There's these activated cells, there's these resting cells, they have different half-lives, and they make different contributions to the number of virions that are in this reservoir. So before ART, by the time you hit chronic infection, it's almost all of these cells that are making the virus. But when you get to treated HIV, it becomes a different story, and this is partly why I'm walking you through this, to take you there. So as you know, there's this two-phase decay of virus in peripheral blood when you put people on antiretroviral therapy. This is a group of people in Uganda who we started on therapy. No, I'm sorry, this is people in Minnesota who we started on therapy, and you can see there's this rapid decay, and then there's this long, slow phase. So, and this has been talked about extensively, the first phase, which is rapid decay, the second phase, which is slow decay. So when you go through and you do the image analysis on these tissues, you find out that the decay in the first phase is elimination of these rapidly dividing activated cells that are making a lot of virus. And so you see rapid reduction and elimination of the predominant cell type making virus, the Ki67 positive. And when you look at the second phase, these are almost exclusively, the cells that are maintaining in this phase are almost exclusively these small resting CD4 T cells that have a much longer half-life. And so the kinetics of the population of viruses that are maintained in the FDC network change over the relatively short time period when you start antiretroviral therapy. I just want to, again, just completely cement this idea because it's going to come up when we talk about persistent replication or persistent virus production. You know, in the untreated, it's these large cells. In the treated, it's these small resting CD4 cells. The other thing that I want to say, and this is, I'm not saying this doesn't happen, I'm just saying we've not seen it. There was, at the early days of the description of this two-phase decay, it was thought that these were productively infected macrophages. And what I'm showing you here is a tissue section from one of these patients in the slow phase of decay. And what you're looking at, the cells stained brown are CD68 positive cells. Those are macrophages. And then you have CD68 negative cells that are actually making virus. And so here, at least in this section, and on all the sections we've looked at, in this phase of the infection on therapy, we don't see productively infected macrophages. And that's just, again, to help make the case that this is the decay of those resting CD4 T cells that contribute to this. Okay. Now, we also have to talk about the FDC pool, and it turns out that that's exactly the same, as you'd expect. You have a rapid decay off the FDC network as those activated cells go away, and then you have a much slower second phase decay, my image didn't show up, of viruses off the FDC network that mirror the depletion of these productively infected cells that are resting. Oh, there it is. So this is a follicle. You can still see there's signal there after 22 days of antiretroviral therapy. But there's something unique here. This is out to 6 months. You'll notice that this graph that looks at the decay off the FDC network goes from 10 to the 4th copies to 10 to the 9th, and that this is the decay you would expect if everybody's going at the same level, but we stop at 6 months. And then you've got some patients that do not decay at the same rate. And so one question is, what does this mean? It's true, but what does it mean? And the second thing is, how persistent is this finding? So this is the first sort of data that we've had that suggests that there's something going on that goes beyond reactivation from latency that is sustaining the infection. What I'm showing you here is data from multiple cohorts of people in chronic infection who've been treated for years. And when we go in and look at, just ask the simple question, are there RNA positive cells in the tissue? We find that 40 to 45% of people consistently have cells that are easily identifiable in the lymphoid tissues, gut or lymph node, that are expressing HIV RNA. And when that occurs, it is almost always at about 10 to the 4th cells per gram. It's a consistent finding. I found it in Ugandan population, people in the US, people in Mexico. It's a very consistent finding. So the mean frequency is about 10 to the 4th cells per gram. Now, all I've told you is that they're making RNA. I haven't told you much more than that. But are they making viral particles? So this is, again, the TSA-ELF, the other form of in situ hybridization. These are actually monkeys suppressed with antiretroviral therapy. And what you see, here you see 2 cells side by side, which is typically what you see as clusters. Here you see 4 cells that have HIV RNA. And here you see a cluster of 4 cells in another cell. And so this is what it looks like. And it's at a frequency of 10 to the 4th cells per gram. So from that, we extrapolate and say that, yes, the cells that are making RNA, but they're also making viral particles that are detected by this technique. So the decay of virus from the FTC net, so those were the frequency of RNA positive cells. Now I'm going to go back to the FTC network and look at the decay rate off of the FTC network. And here's a study that we did and published a couple of years ago now. And we're looking at the FTC RNA, the rate of change, in a group of people, patients, given antiretroviral therapy. They were naive to therapy. We had a lymph node biopsy at day 0, month 1, month 3, and month 6. And you'll see that, and as is the case with humans, not everybody does exactly what you want them to do. So there were a couple of time points variably that were missing from people, or we couldn't find a node or something like that. But variably, most people did what you would expect, that the FTC rate of decay continued to come down, and at least as far as we could tell, about 10 to the 4th copies at 6 months. But there was a subset of patients, excuse me, that had a different pattern. And this was true in the patients that I showed you from the previous study, where there were a couple of patients that did not decay at the same rate. So here's a patient that initially had a decay, but then stabilized. And after 6 months, there was the same amount of virus on the FTC network as before. And here are some patients that had an initial decay and then stabilized. And in every single one of those patients, I know that they were adherent, because we were also looking at drug levels not only in the lymphatic tissue, but in PBMCs every month. And so we know that they were taking their drug. We know that everybody was, these 4 people were undetectable easily by week 4. So they had 5 months where they were not detectable. And yet we could still see in peripheral blood, we could still see RNA on the FTC network. And as I said before, if the input has decreased, then that should decrease as well. This is suggesting that the input hasn't decreased and that there's virus being made somewhere that's attaching to the FTC network. So this is just looking again. You have these small resting CD4 T cells in the part that I didn't mention. If you look here, the morphology of these cells are the small resting phenotype. Now we're just doing the double label experiments now to show that these will be Ki-67 negative. But it looks like the producer, the engine of production that is maintaining this are these small resting CD4 T cells that make small amounts of virus. All right, well, why is this happening? And this is the other, I think, major part of the discussion about persistent production or replication as a model. Some of you have seen this data. In these same patients where we observed this, we looked at intracellular levels of the drugs that they were taking. So tenofovir diphosphate, FTC triphosphate, atazanavir, darunavir, efavirenz. And what we found is, and this is what you're looking at in this graph, is the difference, the percent difference from PBMCs. So if the amount of virus, the amount of drug was the same in the lymph node mononuclear cells or the gut mononuclear cells as it is in the peripheral blood, this should be 1. It should be 0. There should be no difference. But what we found in every compartment, and with the exception of 1 and maybe 2 drugs, is that in the lymph node, the ileum, and the rectum, the ratio was significantly less, and in some cases, the drugs were simply undetectable in these anatomic sites. And this is not novel, this is not new. There are all kinds of examples of antibiotics and other antivirals where you look in the tissues, you see something completely different than what would be predicted in peripheral blood. But so we're calling this suboptimal concentrations of antiretroviral therapy at the site where the virus is actually replicating. So this was just our patients, our 15 patients. We've now got data on well over, I think it's 80 patients that we've studied in the lymph node. And so this is the ratio of lymph node to PBMC for tenofovir diphosphate, FTC triphosphate, atazanavir, which is, I don't think many people use anymore, which is a good thing, darunavir, ritonavir, and efavirenz. And so it just extends the data and adds to the concept that you've got ongoing virus production at a site where the drug is suboptimal. And now this is new data. This was just presented at CROI a few weeks ago, looking at the last class of drugs that we have, integrase inhibitors. And I'm presenting this in a slightly different way. So those of you that are clinicians or ID docs know that we look at inhibitory quotient as a means of efficacy of the drug, and that's the intracellular concentration divided by the protein binding corrected IC90, which I think is probably, well, is a more relevant measure of efficacy. And so this is looking in PBMC. The inhibitory quotient is 5.24. This is what it is in lymph node, ileum, and rectum. So dolutegravir doesn't look so good in the lymphatic tissues. Elvitegravir looks much better. In fact, in the ileum, it well exceeds the inhibitory quotient that you want to achieve. And in the rectum, it at least meets the inhibitory quotient. Reltegravir, we didn't have a chance to look in the ileum, but still, it's not so good. As you would expect. The other thing that we're able to do is measure the rate of decay off of the FDC network, that's our rate of decay, and compare that in those patients to the intracellular concentration of drug. And we found a very significant correlation. So the higher the drug concentration, the faster the rate of decay. And that was true for FTC triphosphate, shown in red, efavirenz, shown in green, and tenofovir diphosphate, shown in black. So another signal that the drug levels are important with respect to virus production, because remember, we're measuring the rate of decay off the FDC network. This is data from the North Carolina group working in collaboration with Ashley Haas in Minnesota, and they're using a new technique for measuring drug concentration that's based on mass spec imaging. And this is actually an HDAC inhibitor, but it makes the point, this is romidepsin, it makes the point again though. Here we've circled in red where you see the highest concentrations of the drug. And then by in situ hybridization for HIV RNA, you're seeing that the positive cells are out here in the parafollicular T cell zone where the drug is not. Where you see it is mostly in the medullary cords of the medulla. And we could have a very interesting conversation about why that is, but I think that's a topic for a different day. But it's just making the point, the drug is here, the infected cells are here. So what happens when you put people on antiretroviral therapy is that while it still is 99.6% of the SIV RNA cells are in lymphoid tissue, the ratio shifts, and it looks like most of this is now occurring in gut-associated lymphoid tissue. And that's another concept that I think we need to think about is on treatment, where are we targeting our therapy? Where do we want to get it to primarily? This was in that paper by Jake Estes, looking at persistent detection of SIV RNA cells before and after antiretroviral therapy. This is before, this is after, and I just want to make a couple of points. You see the expected drop here in lymph node, but you don't see much of a drop in gut, and that's just, again, cementing the idea that the persistent production is shifting to a different anatomic site. But also, and this was something that we had a long, detailed discussion with the people who reviewed this paper, we actually saw an increase in the brain. And so that's the other point that I want to make, is that we have not talked about at all, anybody has, we don't have solid data to understand what kind of contribution the brain is making to the reservoir, and I would maintain the lungs as well, which is a big mucosal area. So I think we need to think about that going forward. So what the origins of recrudescent infection, it's either a single focus that expands from one or two founder populations, or it's this large population of cells that is making virus under suppression, and then you pull the drugs off, and it just explodes everywhere. And which is it? What's the model? So I showed you this viral rebound in 14 days. The predominant view at this comes from one or a few cells. But this is what we saw when we actually looked in the lymphatic tissues when we stopped antiretroviral therapy. So the model for this ATI experiment was that patients had been on therapy for at least 10 years, on average. We stopped therapy. We brought them in on Monday, Wednesday, and Friday and measured plasma HIV RNA. And the moment it was detectable above 20 copies, we got them on the table, took out a lymph node, we did a colonoscopy, and got lymphatic tissue. And what we saw, so when we take out a lymph node, we're sampling 3 times 10 to the minus 4% of lymph node tissue. It's a little tiny bit. Same thing with the gut-associated lymphoid tissue. It's 10 to the minus 5th percent. So when you do your colonoscopy and you're taking pinch biopsies, you've got to understand this issue of sampling. But even with this degree of sampling, we routinely saw multiple sites of recrudescence. So each of these is 3 patients, patient 1, patient 2, patient 3, lymph node, ileum, rectum, ileum, lymph node, rectum, I believe. But the point is, here you have a collection of virus on the FTC network that is exactly what you'd see in chronic infection. It's a huge, massive amount of virus that's on that FTC network. And you have these very hot cells there and in the gut. Here you have big, hot, large activated cells, a large focus of infection here. Now, I haven't proven to you that this is reactivation from one or a few viruses because, there's a couple of days between the time they reactivate the virus and we get them on the table and take the tissue. So we sent peripheral blood and plasma to Brandon Keel and just asked the question using single genome amplification, was this one founder population, which would look like this, from the blood or from the tissues, or is it multiple founders? And the answer in all of these patients, save one, is that it's multiple founders. So there are multiple cells that reactivate at once, or they're making virus under antiretroviral therapy, and you just take the brakes off, and the virus starts. These cells become activated and start replicating. So that's the model that we have and that we think is important, is that you have multifocal recrudescence of virus from multiple founder populations that are likely virus cells that are making virus anyway. So the last thing I want to talk about is this experiment. I showed you these patients, and here they are. Here's this first patient, this guy here, or this individual. And this is day 0. So you're looking at the FTC network. We can measure virus on the FTC network. Here we are at month 1. You've had easily a 1 log reduction, but you come back at month 6. It looks just like he did at baseline. And remember, this particular individual, he was aviremic by the end of week 4, and we did serial monthly checks to make sure that he was adherent to therapy, and he was. So as best as we can do in this kind of controlled experiment, with humans who do what humans do, this is the explanation that we have. This is that second group where you still saw a significant amount of virus on the FTC network, and this is the third group where the B-cell follicles look pretty cold after 6 months of therapy. So I've shown you a lot of data that shows that on therapy, in at least 40 to 50% of patients, and it's probably higher because of the sampling issue, we can find RNA-positive cells. Now, sure, cells make RNA all the time. We can show you that these are mostly virus-producing cells. But you still can make the point of, but can that virus go off? Is it an infectious virus? Can it go off and start a new round of replication? Is that important? So that's one question. But I'd pose the alternative question as well, is even if you've got cells making virus, you're not, as Steve pointed out earlier, you don't know if that's a bad thing. And as he talked about in the Berlin patient, the one difference between the Berlin patient and everybody else is that we stopped the virus from being produced. So I would say that even viral production is an important thing to consider. But we took these patients who you saw in here, and the folks we collaborated with, Steve Walensky and his group did next generation sequencing, so they sequenced the entire genome at 3 time points in peripheral blood, plasma, and the lymph node. And you can see that in these 3 patients, there was clear evidence for sequence evolution, suggesting that there was more than one transfer from one cell to the next. In fact, what we saw was sequence evolution arising in the lymph node that subsequently appeared in the peripheral blood. And that's evidenced by these 3 patients where yellow is the lymph node, and then blue is peripheral blood, here, here, and here. And so there's a lot of debate and discussion about this study and then other studies that are done in peripheral blood. There are differences in the populations that are being studied. There are differences in the way these sequences were generated, et cetera. But again, at least in these patients using next-gen sequencing, we could find evidence for evolution. And the one critique that I agree with on this data is that we need to look out further than 6 months, and that's what we're doing now. We just don't have that data yet. So the alternative view to it's all about latency and it's all about reactivation from one or a few cells from latency. The alternative view, The alternative view is that there's persistent ongoing replication that occurs in most, if not all, HIV-infected people on antiretroviral therapy, and that it's predominantly due to suboptimal concentrations of drugs. We believe this is what maintains the reservoir and that we need better drugs. And those are coming. There are at least 3 drugs that I know of that are in clinical trials or will be soon that are known for concentration in lymphatic tissues. So I think we're going to be able to test this hypothesis in much greater detail once we get our hands on those drugs and can actually look at what happens in lymphatic tissues. So rebound, that's what we're concentrating on. Different sources, different strategies. If it's only latently infected cells, it's the shock and kill or the maintain silence. And if it's ongoing low-level replication, then you have to optimize ART and adjunctive therapies to achieve full suppression. So we started here, the idea of a single focus of infection, a couple of cells reactivating, latency reactivation maintains the reservoir, few cells occasionally reactivated, and we ended here. Now look what we have to contend with, and it's this concept of ongoing low-level production or replication in tissue sanctuaries, suboptimal concentration of ART, so different approaches are needed for the cure if this model is one that I've convinced you is operational. So with that, I will leave you with the words of Shakespeare and thank the very large group of people who actually did all the work. I won't name their names, but just there were a lot of folks, including the patients, that went into generating this data. So with that, I'll stop, and if there's time, take a question.
Hi, Tim. How did you validate that those varying size little green spots or the different ones are virions as opposed to infected cell debris or any number of other possibilities? I'm not convinced those are viral particles.
No, I know. I know you've had this conversation with Ashley before. I would. How can I best answer this? There are a couple of experiments that he's done with cells from the same patient where he has shown transmission of infection, but it doesn't show that those are the same ones. And I think, yes, valid argument. Everything that we can do suggests that those are virus particles. But you're right. They may not be infectious.
No, there are ways to label viral particles now. That there's 50 ways, and then
Agreed.
you just label it your way and show you're labeling viral particles.
Agreed.
I mean, to refer back to 10, 15-year-old papers isn't really good. No. And the in situ PCR was doing all these things, and we don't do it anymore because probably it wasn't really showing us that something that was too valid. So I think you have to be more cautious with that.
Fair point.
I see the evidence that there are RNA-positive cells and virion-positive cells on ART, but I don't see the evidence that you can call that replication.
So
let's look at the possible scenario where that for every million latently infected cells, let's say 2 of those million are reactivating and expressing RNA and virions a week. Those virions are not going to spread in the presence of ART to other cells because the antiviral drugs block the new infection but don't block expression in a latently infected cell. And you're never going to measure a decline in the reservoir at such a low level of reactivation because the half-life of those reservoirs would be 50,000 weeks or 5,000 weeks, way too long to be able to detect at a low level of replication. Yet, when you remove antiviral drugs, those 2 per million per week are going to very quickly ignite a forest fire and have virus spread all through the system.
Yeah.
So again, I see the evidence for a small percent of cells making RNA and virions in the presence of ART, but I don't see the evidence for replication.
So the evidence for replication comes from the sequencing paper, and that's the limit of what we've got. And go ahead.
And that paper from what's his name in Chicago is pretty controversial, right? There are people that disagree with that result or not?
Well, sure. There are a lot of people that disagree with it. If you just look at the letters that made it into Nature that were the response to it, I think there was one published, but more written. It needs to play out. I mean, other people have to do this. We have to see if we can replicate it. We have to see if we get that same result if we go out further than 6 months. I think there's a lot of what ifs. But the point that I'm trying to make is that if there's sufficient evidence to say that we need to think about this and we need to study this more, the reason for doing that is because it's going to give us a different pathway to the cure. We have to think about how to fully suppress this if this is true. And so that's the one point. But then the other point is, if these cells are making RNA, and even if they just go on to infect one other cell, you're still probably inciting an inflammatory response that I think is pretty important. And then the third thing is that, when I'm saying our evidence for the decrease in the reservoir, I'm not talking about frequency of productively infected cells, although I mentioned that. I'm talking about the decay of virus off the FDC network. That's the difference. Up here?
Hello?
Yeah.
So, did you isolate the virus from those virus-producing cells just to look for resistance mutations? Because maybe those cells are still producing virus not due to be optimal drug concentration. Maybe just because these cells are infected of some resistant viruses. So it's still producing because the drug is not gonna affect them.
So there's two ways to respond to that. One is when we put people back on therapy, after the virus came back, we had sufficient virus in the plasma to measure for resistance mutation, and there were none that emerged. They weren't there to begin with. But the second thing is, again, if you look at that paper by Lorenzo Redondo, the model that they have there is that the reason you don't see emergence of resistance, or at least we don't detect it in the tissues, is because you have low-level replication. The frequency of infected cell is producing virus is very small, and so there's no fitness advantage to the virus to mutate and become resistant. At least that's the model that they put forward.
Are those measurements also worthy to defective proviruses?
Say that again?
Are those measurements of messenger RNA worthy to defective proviruses? It's not producing a perfect virus. It's producing
nobody, to my knowledge, has actually gone in and what I was talking about is they modeled the data and said that you're unlikely to see mutations because of the low level of production. I'm not aware that anybody's gone and actually looked.
There's, I think, two posters on CROI this year showing lymph node biopsy and sequencing compared to peripheral blood and showing that the viruses were basically the same, sequence-wise.
Sorry, whose data is that?
I don't remember, but I saw that, and people also replicating that in other labs.
Yeah, so again...
Also compared to full genome sequencing.
Right. This is the debate that occurs with the people who do the sequencing and the evolutionary biologists. I think the data that you're referring to is probably the paper that Dr. Kearney presented recently at CROI.
Yes.
Different population entirely, different sequencing entirely. And I know that the groups that actually know what they're talking about with sequencing, they all disagree with each other. That's why I say this is a question that's in evolution, so to speak. And there needs to be more data. There needs to be more people looking at this in order to get to the truth about that particular question.
Any other questions? Thank you, Tim.
Yeah, thank you.